Maybe this will work. Is this working? Oh my gosh, I think it worked. I think it might have worked. Maybe worked. Okay. Oh, there's a delay on the YouTube stream. This is going to be the best start of any video, and no one's going to watch it ever again. There's a delay, and so I can see myself looking very concernedly at my computer. Yes, it worked. Okay. Well, hello everyone. What a disaster. Welcome to live. I'm just going to pretend like this is super normal. Thank you, everyone. You're amazing. I am computer illiterate. Okay, yes, Ryan Gordon, you get to take full credit for this. Hello, everyone. You're friggin' amazing. Okay, let's go ahead and get this running. Um, yeah, screw it. We're gonna roll with it. Guha, you've been amazing. Let's jump right in. All right, all right. Okay, so we're gonna pretend to go live right now, and then I will introduce you again. Sorry, or thank you to everyone. Hello, everyone, and what the hell is up with vaccines and COVID and antibodies, and what are these things? Why aren't they here yet? What are we doing as scientists to make that happen? My name is Susanna Harris, and I host Ask Me I'm a Scientist so that every episode I can bring on a new expert who tells me about what they're working on, and I don't have to do all this Googling and being half wrong all the time. I'm super excited for this week's guest because, first off, he's amazing. You've probably seen me slip up in about 10 versions of this video, but more importantly, the reason that he's here is that he's going to be talking with us about his research in antibodies and developing them for viruses and how that's related to COVID right now. Anyway, Guha Arunkumar is a PhD candidate in microbiology at the Icahn School of Medicine at Mount Sinai, and he started working in a similar field, definitely viruses and vaccines and stuff, but his whole life has sort of turned upside down, and for our betterment, because we're about to learn some really cool stuff. Guha, hello for the 18th time. You're amazing. How are you doing?
Good, good. I'm glad we're back on. I'm glad that things are working right now.
Yeah.
This has got a lot of fun.
Well, you have been amazing. As people know who have hopped on this show before, it is a live show, and takes a lot of bravery for people to hop on, and it's actually just more fun for me to watch myself fail. So enough of the backlog. Anyway, so what has happened with you in the last 2 months or so?
Yeah, all right. So essentially, the way that the story goes is that I am currently in the fourth year of my PhD. Actually, nearing the end of the fourth year of my PhD, which is not very pleasant when I say it out like that. I mostly work on antibodies and vaccines towards until a couple of months ago. About in January or so, my boss decided that us as a lab that focuses on antibodies, generally speaking, and we like to study viruses and how we make responses against viruses, and how you can test for those responses against viruses, especially when someone falls sick and recovers. Having all of these areas of interest towards viruses and the proteins on their surfaces in general, we decided to pivot towards COVID vaccine research, or like just basically COVID-related research, not necessarily even from a vaccine standpoint. And the idea was to produce and generate as many reagents and tools as possible in order to be able to study the virus. So that started somewhere in about January or February, and then over the last month and a half, it's been going on in full swing. There are a couple of other lab mates and colleagues of mine who've basically taken the lead on this in terms of a few projects where they're either directly working with the virus or they're directly trying to understand, when someone recovers, how exactly do they produce antibodies or what's going on. And a lot of us in the lab, along with those people, are trying to help the hospital set up a test, etc. So it's been pretty chaotic, to put it mildly, but it's also good in a way that we're able to help to whatever capacity that we can. And some of us are fortunate in being able to live close by to work, to be able to come in without having that higher risk of sort of falling ill or something like that.
Yeah, well, I mean, thank you so much for everything you're doing. And science is such a collaborative effort, and it's amazing to see how people can just jump on, switch topics. You're pretty far along in your PhD, and in their chat, so many people stayed with us. I can't thank them enough. Jack Yuanwei Chang says, seeing that power outlet makes me miss my lab. So you are one of the few people who has kept their current PhD research going, kind of. It's totally different stuff. Um, but how has that transition been? You know, how did you transition from the one virus to the ones you're working on now? Is there a lot of overlap, or are you just having to like start from scratch?
So it's one of those things. So since I'm in a department of microbiology, and although we say that a lot of the people here mostly work on viruses, so what's been really interesting and kind of exciting to see is that each lab in their own way has sort of pivoted their area of expertise towards this virus, right?
Hmm.
So you have people who study herpes viruses, HIV, like influenza, a lot of influenza work that goes on, and also other viruses like dengue, West Nile, and all of these things. And each lab has their area of expertise where sometimes they look at why people don't fall sick, like why are their immune systems really strong, or some people look at how can we make vaccines, and we look at like studying the antibody responses and all of those things, right? So everybody at some level was sort of equipped to be able to transition their work and sort of reappropriate all of their apparatus and reagents, etc., to be able to generate and bring their expertise to the table, whether it be engineering new viruses that can be easy to study, or whether it be trying to come up with vaccines or trying to test drugs that are already out there. So I think in that aspect, we're a bit fortunate. This would be a whole lot harder to do if you were a lab that's, say, focused on cancer biology and then you had to start from scratch. But since we're used to working with viruses, specifically RNA viruses in this department, and this virus, the SARS-CoV-2, falls into that family, and that worked really well for us.
I mean, that's awesome. And you mentioned something, RNA viruses. So, from what I understand, it's been probably 6 years since I've taken a virology class, but different viruses are sort of classified by their genetic material, right? And so there's RNA, there's DNA. Why does it matter? Isn't a virus just a virus? Like, why? Originally, we were talking about, isn't this like the flu? And I know it's not like the flu. Um, but why does it matter if we're specific about these things? Why can't just every vaccine work on every virus?
Yeah, so the interesting aspect of it is that you have all of these viruses with different genomes, right? Their DNA is different or the RNA is different. Some of them are single-stranded, some of them are double-stranded. Some of them might be that if you have a piece of RNA, which is the genetic material, and it's a set of letters, that can directly become protein. So as one of the people on one of the earlier episodes pointed out, like that's the central dogma, right? Like basically you go from DNA to RNA to making protein. So the cool aspect of it is that viruses are like incredibly small compared to how complex we
Hmm.
are, and they're capable of doing all of these different things, which are versatile and having maybe like 8 genes, but they are capable of coding, let's say, 15 to 20 different proteins. So it's always really important to know the nature of the virus that you're working with, especially in terms of how the DNA or the RNA is, because that might influence what you might target, and that might influence how likely it is to change. So let's say, for instance, if you had a really big virus, um, the virus might have co-opted mechanisms through the course of evolution where if it makes copies of itself, it's capable of doing it in a very like stable way, or it's capable of doing it in a very good way without having multiple errors that are introduced into the genome. And then if you have an RNA virus, or if you have a virus like flu, then when it makes multiple copies, you have differences that are introduced each time, which lead to mutations. And when you have mutations, that's kind of why the virus changes every year, basically. So in that aspect, it's certainly important to know or worry about the classification, the nature of the genome, and what these proteins functions might be.
Yeah, so I mean, there's so many different aspects going on here. And from what I understand, your specific research is looking at these antibodies and, these specific proteins that are really important for distinguishing these different viruses. Kind of what's the main technique you use to try to create antibodies or create a vaccine? Like what I'm realizing right now how little I know about this topic.
No, I mean, I think this works really well, right? Because I feel like constantly we always pigeonhole ourselves into our areas of research. So it's always nice to hear about, and I learned a lot from some of the previous talks that you've had over here
Thank
too.
you.
Though one of the things to keep in mind is when you talk about a virus like flu, for instance, right? you see the virus as it is. So let's imagine that this is the virus, and then you have things sticking out at the surface of the virus, which are these proteins on the surface. Your body will look at that, and your body will make an immune response against that. That immune response might be in the form of antibodies, which are created, and they are like these Y-shaped molecules, which then go bind to these viruses. And the idea is when you have these antibodies, when you see the same virus or when you see something similar, you may be able to recognize it all over again, right? So these are called surface glycoproteins, and the reason they're called that is because they're on the surface of the virus, and they're glycoproteins, which means they're proteins, but they have some sugar on them, or they have some glycan groups on them that have been added through the course of it. And the reason is because they hijack our machinery, they go through the same processing that your proteins would go through. So when they get taken out of the cell, they have some of the things that some of your proteins would have.
So wait, hold on.
And these,
okay.
yeah.
So no, you're fine. So glycoproteins, like basically what I'm understanding is sort of the same way as when you're recognizing a person, or let's use a dog. You're recognizing a dog and you might say, okay, it has this color fur, it has this color of eyes, it's this tall, it's this short. So I know what kind of dog this is. Is that sort of like the proteins are these characteristics that tell your body what that is and if they've seen it again?
Yeah, essentially. So it's basically what you might say is like one of the first or the most defining characteristics of a virus because your immune system recognizes it first. So imagine there's like a virus that's floating in your body or in your lungs or outside of your cells. And if your immune system is able to see that and say, hey, I've seen this before, or this looks weird and I haven't noticed this before, then it'll either recognize it because it's very, familiar to it, or there might be processes through which it'll start recognizing something that's unfamiliar or foreign and that shouldn't be there. And in doing so, then you make some sort of response towards it, and that could be an antibody response or something else.
Huh. Very cool. Yeah. So
at least for the COVID, as I think someone mentioned earlier, like you have those spike proteins which are on the surface of the virus, right? So these are things that like jut out of the surface, which kind of give it that crown-like look when you look at it under an electron microscope. That's why it's called a coronavirus, because it has a crown-like appearance.
right. It's just beautiful. I love electron microscope stuff. And so it sounds like a lot of the stuff you're doing is really like deep biochemical assays. Is that about right?
Yeah, we've over time figured out ways to look at antibodies, whether people produce antibodies or not, and we've also figured out ways to make antibodies, which we can make in either through humans or through mice, or we look at like nature has already done the work. So you take someone who's recovered from a disease, and then you see what antibodies that their body is produced, and which of them might be good or which of them might be bad.
Okay, that makes a lot of sense. So yeah, going back to the dog, you'd be like, "Oh, that's a nice dog. I've met this kind of dog before. I liked it." Versus that dog bit me, and your body kind of
Yeah,
remembers that.
basically.
So okay, I want to launch into all these questions because we got some on Twitter, but
Yeah.
I mean, I'm going to hold this up to the camera. Let me see. I have to switch over my views, but I'm going to hold this up to the camera so people can see just how many questions we've got. I'm going to try to get to as many as we can. Um,
It's the season.
Oh man. Okay, so but one of the questions that I think came up more than all of the other ones is this. So A underscore Ajaswi. I of course picked the hardest one to pronounce. There's like 16 of these questions. I picked that one. Anyway, it says, can a person who recovers from COVID-19 get it again? This is probably the question we're all asking.
Yeah, so this is a fantastic question. And I think one of the concerns of us as a part of the public is like, oh, you hear of all of these reports and you read some of these news articles of people of reinfection and all of those things, right? So there are a few different factors. There are a couple of hills that I might die on over here. But the point being that, so if someone gets infected by the virus and they make an antibody response, like a really good antibody response, and what I mean by good is they produce antibodies which, if they were to see the virus again, it would go bind to the virus and prevent the virus from entering the cell and making more copies of it. So those are like neutralizing antibodies. Are they capable of neutralizing the virus or neutralizing the threat once they see it? So if someone were to make these antibodies, then their likelihood of getting reinfected is very low. When you talk about people who recover, there's a possibility that they might have recovered without an antibody response. So there are other aspects of your immune system, like you have your natural defenses, which play a role. So if someone tests positive, but they're not very sick, and they don't get very sick, it's possible that their immune system in different aspects, like there are these cells called T cells, there are other cells that look at a virus, they break it up into pieces, show it to your immune system, and then they say, hey, look, here's something that's foreign. They're called antigen presenting cells. So there are all these different types of cells in your immune system that might play a very good role in controlling it. So it's always a case, and there are some studies now which are showing it because we have the luxury to look at that right now over time, and even some studies out of China where they've controlled the numbers, looking at whether people have produced antibodies. So if you don't produce antibodies, there are other ways you can control the virus.
Okay, so wait.
That means that there is a
Yeah,
chance.
so
Sorry.
no, you're okay. So like I just go back to this. So people might not know this. I have a bachelor's of science in microbiology. I actually just got a PhD in microbiology. The one class that I have ever, well,
Congratulations.
wait for it. The one class that I have ever dropped is immunology because of honestly, all of these different words, and they're so complex, they're all changing. So
Mhm.
I just want to make sure that we kind of have a working knowledge. So antibodies are produced by the body, by humans, by mammals. Let's not get into that. Um, they're produced, and they're kind of a way for us to, once we recognize a virus, to kind of attach the virus, make sure that that little
Yep.
key that it has to get into our cells is blocked off. It can't find a lock anymore. Okay, and then different immune cells, though, that's not our only way of protection against these viruses. There's also these other mechanisms, which is great, but it sounds like if we use those other mechanisms, if our body uses these skills to get rid of the virus, we then won't have that long-term immunity. Is that right?
So
Okay.
if you don't fall, let's just say that like your body, even if you get exposed to the virus, but like you don't necessarily make antibodies, and you're like you said, you're able to control it. There are a couple of those arms of the immune system. Specifically, there are some cells called T cells. They also have a memory component to them, and the idea is even those would be capable of recognizing a virus that they've seen before. So there are ways, and even if you look at things like mucous membranes or our skin, these are all considered to be a part of our defense, right? So if we're able to control it due to some of these factors to start with, and some of it's called an innate immune system, which means it's a very quick initial non-specific response. So let's say that we're able to control it because your body gets inflamed, you get a fever, then it's possible that like there's no memory, there might not be a memory aspect to it. So there is a chance that you might get
Mm,
reinfected. Um, yeah, so that's basically how I would put it.
yeah.
So there are different arms of the immune system, and there are memory components to some and not to some.
Okay. And that leads us right into all of this stuff where, like, let's say someone does make a bunch of antibodies. We've got questions. Let's see, from Team Olive. Can we transfer these antibodies from person to person by blood? We also have questions people are asking, and maybe it's related, maybe it's totally not. But Anso Bach says, would it be like for the flu, one shot every year? So if we just took antibodies from somebody, or if we just had a really simple vaccine, um, is this something that's going to be easy to solve? We just take blood from somebody and we put it in somebody else?
Yeah, so that's really great, and that's actually something that we're doing
Yay.
over here at Mount Sinai in New York, and it's approved at multiple centers in the U.S. and in other places in terms of compassionate use because these are like it's an emergency condition, and we're trying to do everything that we can. So if you can screen people who've recovered and see who has high levels of antibodies, if they're willing to like go to a blood donation center and donate their plasma, where you basically take their blood and then spin it down, and you specifically get some of the immune components to it, like the antibodies. You can transfer the plasma provided there's a match. You would look at all of those things. You can transfer the plasma into someone else who's possibly, let's say that they're an older individual and they're not capable of producing antibodies because your antibody response or some aspects of your immune system become slightly weaker as you grow older, or you have some sort of a immune condition that prevents you from making a good antibody response. Then provided that there's a match and that they have good levels of antibodies in their body, you can actually transfer it, and that might potentially work. And that's currently approved in terms of an emergency compassionate use, and we already
Mm.
have patients here at Mount Sinai who've been transfused with plasma.
And is it working?
Yeah, so there was one report that came out of China, which essentially looked at 5 patients, I think, if I remember correctly, which said that we've done convalescent plasma transfer, what the plasma transfer is called, and said that it helps for sure. Considering the state of affairs and considering the fact that like it's a bit of a desperate situation, it would still be worth the effort as long as you can show that it doesn't get worse. So even if you have no improvement
Mm.
to an improvement, that's considered a win as long as there's no deterioration that accompanies that, basically. And you would have to basically, we've designed here specifically, we've designed an assay to test to see whether people have these antibodies or not.
Wow. Okay. All right. Well, I guess it makes sense. And you said the word compassionate treatment, which is basically like you're trying whatever you can,
Yeah.
and because it's such a dire situation. Is this something like, do people have to get this every day? What's the difference between getting antibodies and getting a vaccine?
Yeah, so if you talk generally, not specific for this, antibodies, like if you had to produce them, then they are in general expensive, right? So the idea with vaccines is like you can manufacture it depending on what platform of vaccine you use, and there are many different platforms. You can do it in a preventive way where you can give someone the vaccine and they will have a response and then they'll be protected over the course of time. And those are generally like preventive or prophylactic vaccines. And then the case with antibodies would be that you might give it to them if they're sick in a therapeutic way, right? But in general, antibodies are like expensive to manufacture. So currently, it's one of those things where you rely on plasma
Mm,
donors and transfusions. And I'm personally familiar with a couple of cases where they've required multiple transfusions. So they've required maybe 2 to 3 different plasma donors.
oof.
And that will be definitely subject to whether it improves them or not. I again think that's based on like you would not wing it, but you would determine the course of plasma transfusions as through the course of it. But that's more on the clinical aspect of it, and I'm definitely not an expert when it comes to that.
Hmm. That's so interesting. Yeah, so the
Yeah.
vaccine is like you're going to make your own. Well, I mean, actually, this is the next question, of course. I don't know. Let's see. Wow, people are interesting. Um, I want to find somebody. Okay, uh, you know, we've gotten the question a bunch of is this going to learn? Is this going to turn into long-term resistance against the virus? Is one vaccine going to be enough? Somewhere up here, yeah. So Ansobach said, will it be like the flu, one shot every year?
Yeah.
Yeah, we think.
Yeah, no, sorry for not answering that
No,
earlier.
okay.
So it's one of those things where I think there are a few subject experts who believe that this might become um, like something that circulates year in and year out. Whether we will see a seasonal trend like flu, where flu peaks in winters, for instance, whether we will see that or not is unsure, right? But the one thing that we should know about this virus is that like it does mutate, and there are numbers which show this. It mutates a lot less than flu, for instance, right? So flu changes at very rapid rates every year, which is why you have a new vaccine that's created every year. They use the same way to do it every year, but then they just decide which strains go in. As for this, when they've looked at viruses previously that are related to this virus, so when they've looked at the Middle Eastern Respiratory Syndrome coronavirus and SARS, they have found that over a year or two, your immunity tends to wane after a year or two. So if you make antibodies, they might stick around for a year or two, but after that, there's a chance that they might not be good if you got re-exposed um, to the previous virus, right? But the idea is that if we're able to control it and then make sure that enough people are vaccinated and you have a concept of herd immunity, then this may not become an annual thing. But that would be the case if you had a vaccine that works. And as for whether it's one vaccine or not, again, we're in that situation where I think there are above 80 to 90 vaccine proposals and candidates, 2 or 3 of which are already in the clinic. So
Wow.
they're already testing it in humans, and there are different phases to it. And it's a matter of throwing everything against the wall and seeing what sticks. And it's kind of nice to see that there are a lot of people getting involved because vaccines are not necessarily profitable, and it's always nice to see the fact that whether you're industry or pharma or anything, you're getting involved in the process right now.
Sure. And, going back to what we were talking about at the beginning, where you have changed your trajectory very quickly and you've changed your major topic, but there's some similarities. But it also, I think this highlights, at least for me, that not all viruses are the same. And, we can't necessarily say, oh, the flu is like this, so this is what we're going to see. Or like SARS,
Yeah.
the original, I don't know what that means, like
The OG SARS is how people
oh
call it. Yeah.
gosh, okay, yeah, version 1.0,
Yeah.
or MERS, you know, and even things like herd immunity that I think that we toss around the words herd immunity um, very, very openly because it is something that we know something about. But from your opinion as a virologist, is it too early to just kind of predict what is going to happen, or should we just say we know all this stuff, let's make a guess and go with it?
So I think it would be very bold for anybody to come out and say that. And we've seen that happen a string of times with experts, epidemiologists, virologists, all of these people, right? Where they've said like, oh, you'll have a 30 to 70% hit rate, and that, in certain places may or may not have been true. But there are so many measures that come into play that's just not scientific, like not directly scientific or not directly molecular, right? So even when you talk about social distancing and stuff like that, there are so many aspects to it that we could do and we could take, which cannot exactly help you directly determine and say, okay, you know what, in a year from now, we're not going to have to worry about this at all. I think it would be very bold to be making such definitive statements, but that's just my personal opinion. So it's better to just look at the candidates that you have, try to put in money in terms of research. And one of the issues with SARS, for instance, was I think SARS vaccine development at some level halted because the money was pulled, because SARS came on really hot and then it just disappeared. Like it just went away.
Yeah.
And you don't want a situation like that happening right now.
Right, absolutely. I actually really love this question. In Instagram, Sheldon, yes, woo, says, "Sorry if this sounds dumb, but does having the recent flu vaccine improve your chances at all?" And I really appreciate this question because I think it's a valid question. I think it's something that people are nervous about asking. So yeah, if I've had the flu vaccine, if I've had chickenpox vaccine, are there any vaccines like that are going to protect me already?
So technically speaking, no. If you talk about it, when you talk about direct, like making responses against a vaccine and a virus, I would say no. That wouldn't be the case. The one thing that is very important, especially when people were talking about getting the flu vaccine back when this was, let's say, early January or February, right? If the overall effort, if we, a lot of us got our flu shots, and that may not necessarily be the case, but if you got your flu shot, then you're less likely to overwhelm, or you're less likely to even like weigh in on the healthcare and their burden, basically, right? So you're not
Mm,
adding to the already existing healthcare burden. And that's why I personally think like it has value. There's absolutely no doubt. Everybody should get their flu shots if possible. And like I say, this also working on it. It's just one of those things where it's more from the standpoint of like we don't add on when there's an already existing problem, because if you got your flu shot, you're more likely to not have a severe episode of flu, or get a secondary bacterial infection, because you wouldn't be getting flu in the first place, depending on the season and the vaccine. Um, there are some reports which suggest, and this is anecdotal and yet being verified, so I can't necessarily comment on it with confidence, that like if you get a vaccine like the BCG vaccine, then there's a chance that you might, which is for tuberculosis and happens in several countries, like India, for instance, then that might be beneficial at some level in terms of providing like a non-specific immunity. So what that means is like if you get the vaccine, that some aspects of your immune system might be a little bit on more high alert. I think there are a lot of studies that are being right now to validate that, but it's that's a bit up in the air. In my personal opinion, I'm not.
no, I mean,
That's the way I would.
I think that's great. And I love the part that you put in there of maybe there's not ways that we can fight COVID right now directly with a vaccine, but what we can do is we can support the healthcare systems. We can do things, we can
Yeah,
keep ourselves safe. And I just love that that's also your take as a virologist. Uh, okay. Let's see. Oh, okay. I think this will be the last one. I don't know, but and by the way, there's a really interesting conversation happening in the chat over here.
live chat.
Yeah, and so I mean, a lot of people know way more than I do. Okay, actually, I do want to ask.
I'm pretty sure it's also more than I do, because like I think the extent to which we're consuming this information, like it's pretty great, and there's a lot of openness in terms of communication, so that's pretty good.
Yeah, it's great. So let's do one there and one more from Instagram. So Akshan
Sure.
Garayev says, "Can quote immune people still spread the virus? Do we know this yet? We know that low levels, if someone is a carrier and they are currently infected and producing the virus, that they can spread it. But what about somebody who already got over it? If they get reinfected or if they have like low levels, are we worried about them reinfecting? Is this going to be an issue in the future?"
Yeah, so this was me thinking about it, and this is again definitely more of a personal opinion. But the way I see it is, if you're someone who's basically been infected and you've recovered from it, and let's say that you have a good antibody response and you're able to control, or like some aspects of your immune system, whatever it might be, and you're able to control infection, the virus would not reach like a mass or a critical mass at which point you might be shedding it to other people.
Mm,
So I'm not saying that you would be a giant might, because what would happen is you could still touch a door which has the virus on it. You could go touch another door which has, which is like someone else comes and touches it. You can touch your face after that. You might not fall sick, but then you're acting as someone who could potentially transfer it from one surface to another. As for whether you would shed it yourself is definitely unclear. And I think that goes back to the fact that we don't know much about this virus because it's like the new kid on the block. And but I would have to say that if you have recovered and if you do generally have a good response, you might not allow it to reach the stage when it infects you such that you might be shedding to other people around you.
Huh.
Or at least that's what I would hope for, because that makes life a lot better for a lot of us.
Yeah. Yeah. Okay. So, um, last question I'm going to ask, other than my closing question. Frank
Mhm.
DeCisi says, does the use of supercomputers help expedite the development of a vaccine? Yeah. So, other than the biochemical aspects of these things, are you using computational approaches? Are there other groups that are kind of working on different sides of this problem?
Yeah, so this is really cool. I think there should be like a very good coming together of topics like AI, for instance, along with drug design or vaccine design, right? So there are some people who do it for flu. So what they do is like they look at 10 different viruses and then they're like, okay, all of these are different. Can we find one in the middle which is very similar to all 10 of them? So if we use that, then maybe you can make a response against all of them, or that will make sure you don't get severe disease to all of them. Um, Currently speaking, when you look at COVID-related stuff, there are people who are trying to use similar, like I think this was my uncle sent this article because he works at IBM and this was from IBM. But it was about repurposing computational capacity to look at interactions of, let's say, if you have one protein and it's trying to bind to a cell, can we look at that interaction and where it interacts and how it binds and try to find things that could disrupt that? So that's more of like, I want to say drug design rather than vaccine
Yeah.
design. And there's a very heavy computational aspect to targeted drug design, and that's, I think, getting better over the years, and that's currently happening right now. There's also an excellent study that came out of these like 20, 30, 40 different biologists. It's a very star-studded lineup, and it's from Nevin Krogan's lab. And basically, if you look at it, they've done a lot of computational work where they take a virus, they take cells, and then they just let them sit together. And then they figure out what are all of the proteins in the cell that all of the viral proteins interact with. And they make these beautiful
Hmm.
networks, which is all computationally generated. And then they figure out off of these, which ones can we target? Do we have drugs that are approved, like hydroxychloroquine or something like that? And can we maybe just repurpose the drug, which might be for cancer treatment or something else for that? So that's definitely happening, and there are some really, really cool studies out there which show that essentially.
That's so cool. I literally just got goosebumps because I think this is what's really beautiful about science. And this entire situation is so chaotic and so just devastating. And I think it's important to keep an eye out for some of the bright sides. And I think, like you said, there's
Mhm.
usually not funding for vaccines, which to answer a couple of the
Yeah.
questions, I will go ahead and answer this question. You can follow up. But yes, I do believe vaccines work because they do work, and you should get them if your doctor suggests it. And I will die on that hill. Thank you. How about yourself? Do you? Yes,
Nice.
please.
Yeah, sorry. No, so this topic hits very close to home, and I mean, I got into vaccine research mainly for a lot of these things. I envision a career where I'm in vaccine research and development, and I absolutely love this aspect of it, right? Um, there are a lot of people around who can't get vaccines, and there are a lot of people around who, even if they got it, it wouldn't do anything for them because they're probably immune compromised, they're probably immune suppressed, they've probably gotten a transplant and they're on medication, they have some sort of autoimmune diseases. And us by getting around them as comparatively healthier individuals who might not have an underlying condition can play our role. And this goes back to the whole talk about herd immunity, right? So yes, if there's a vaccine, I absolutely think that people should get it, whatever it might be for. Even if it's not a great vaccine, which is the case with flu sometimes, and we're endlessly trying to make better vaccines for that too. You should get it because there are people around you who simply can't, and
Yes.
it's the least you could do in terms of protecting them, basically.
Absolutely. I love that kind of set of values, and I think that you've repeated it a lot of different times in terms of, taking care of yourself so that other people have more access to care. And I really appreciate that you brought up that point about people being immunocompromised, people unable to get the vaccine. That, of course, we're not saying, and neither of us are saying like everyone should get this no matter what. We're
Yeah.
not giving medical advice, but rather if you're told that you should get a vaccine by a doctor, by your healthcare professional. There's a reason for that, and you're saving other people's lives. Just because you're young and healthy doesn't mean that your decision doesn't affect the people around you. So, Guha, there's one question I always ask at the end, which is, what are you excited about in this field? What are you actively learning? What gets you jazzed to come into work? Anything like that?
Yeah, so like I said, I love working on viruses. I love vaccine research, and I think it's very translational. So the idea of translational research is going from the lab bench to the bedside, right? Is able to being able to translate your research to make it to the clinic, for instance. That's kind of what keeps me going. And the coolest aspect of it right now, and through Twitter and other platforms, I can like there's just a lot of information that's being consumed. The part that's really cool to me is that a lot of the vaccines that are currently either preclinical or being tested or are proposed are very innovative vaccine platforms,
Mm,
right? So you're moving along and you're innovating as you go. And the idea is some of these platforms are so amenable to just switching. So, like, if you have a new virus that pops up, you did it for Ebola. If you have a new virus that pops up now with SARS-CoV-2, you can just reappropriate it towards that. And then you can do the same for the next, virus that pops up,
mhm.
because this probably will be a recurring event in terms of us encountering new viruses. So that aspect of it is definitely really appealing to me, that there's a lot of beautiful engineering and creativity and just like platform vaccines that are working or that are being looked at right now, which can very easily be reappropriated and given to developing countries, easily be produced, scalable, cheap, and accessibility goes up if that's the case. So it's really cool to see some of the work that a lot of people are doing all over the world right now.
That's it's just a great perspective. And, thank you again so much for what you're doing. I know that there's a lot of
Thank
scientists
you.
who are, frustrated. There's a lot of people who are frustrated that they can't go in and do their work. But I think there's a huge, I mean, you're taking on a really big responsibility, and you're in a place that's a difficult place to go out. So, thank you for everything you're doing. Thank you so much for taking the time to do this.
No, thank you. Yeah, thank you for having me on.
Absolutely. So we've got your contact information underneath your photo, which you're going to see the video, and
Sure.
your photo or your video keeps like expanding and contracting. So it's keeping me on the ball, which is great. I think that's just
Yeah.
the theme for this whole thing, but 100% worth it. This was awesome. So are people open to, can they send you a message on Twitter, Instagram, ask some of
Oh,
these
yeah,
questions?
absolutely. Yeah.
Cool.
No, I'm more than happy to talk about anything related to this or otherwise too. This, I'm definitely, it will be a good time.
Absolutely. Well, you are in high demand both in the lab and out in the public. Thank you again so much. And I hope you get a break from lab soon.
Yeah, yeah. Thank you so much. Yeah, hopefully too. Take care.
Thanks. Okay, everyone, thank you so much for your patience. That was quite the adventure if you saw the other two takes, but now you know for sure that we are doing this live. And it's worth it because scientists are the coolest people, and I love talking with all of you. I love seeing these questions come in. Please, to keep me going, like, subscribe, share it with other people. Let me know what I could be doing better. There's always room for improvement. And also, definitely send me along ideas of people that I should be having these discussions with, whether it's COVID, human health, astronomy, I don't care. As long as it's science, as long as they're an expert, I'm very excited to talk to them. So until next episode, my name is Susanna Harris, and I will see you very soon. All right, bye.